So this year's Copley Medal winner, which I think is the world's most prestigious medal, it's certainly the world's oldest scientific prize, has gone to Gregory Winter. For basically creating therapeutic antibodies, what's called monoclonal antibodies. And what he did was the thing the Copley Medal has actually gone to is what are called humanized antibodies. But I think he also has done this outside species using test tubes as well. So which is called in vitro. In animals, it's in animals it's in vivo. That's what we call it. So what historically, before he came along, if you wanted to grow antibodies, you say grow them in a mouse or a rabbit or something. If you then took those antibodies, which are maybe against polio, against meningitis, and you put them into a human, our immune systems would recognize them as being foreign proteins, so it would destroy them. So you get a bad reaction, it might even kill the person. What he did was he took an antibody as this shape. Well, this is actually joined by bits. This is an antibody shape, okay? It's a Y shape. So, protein, okay, this is normal. Okay? What I should have done was black actually, better. So what he did was so that part of it, this end actually binds foreign particles, okay? And this binds things like macrophages and natural killer cells. It binds. So what Gregory Winter did, he took an antibody and he humanized it by, so the black bits are mouse and the red bits are human. Okay, this is diagrammatic, but it's just to illustrate kind of what he did. And then you have, say, it's just diagrammatic. So the red bits are mouse, the red bits are human, and the black bits are mouse. And so when he engineered this and grew them in the mice, if he then stuck them into humans, they would work and they would actually help our immune systems, and our immune system would make more copies of them. So this is kind of an outline of what he did. This is partially humanized, he then fully humanized them. Okay, so phage displays are when you have phages of viruses that attack bacteria. This phage display is when you have millions of billions of phages attached to the solid surface. So these are used to identify protein, peptide, protein, and protein DNA interactions, okay? And what Gregory Winter did was he took a gene of interest. By the way, the phages that are used in this are things like M13, FD, the T4 phage, T7 phage, and lambda phage, okay? So what he did was in the phage display, he took a gene of a protein of interest, okay? So, gene of, okay, and he put it into a phage protein, phage coat protein. So you get basically the phage like this, that's the capsid, okay? And you have the gene, the piece of the phage DNA RNA, and you put the gene of interest in that, okay? So then that then gets expressed from the outside of the phage. This is the phage code, okay? So what this does is you have, that's the phenotype, and that's the genotype. So this basically, his work showed a connection between the phenotype and the genotype. So once you had all these millions of phages displaying, billions of phages displaying a protein, he didn't screen this. And by washing over millions of other bits of protein antibodies to detect interactions between, and so when an antibody, when the humanized antibody lands on the protein of interest, then you've identified the antibody, and then you can amplify. You can amplify it. Okay, so once you've identified, and then you can amplify up. Okay, I think it should be an I there. Amplify. Okay, this basically led to the creation of antibodies, which are used throughout medicine universally. So this is sort of an overview of what his work did that got the Copley medal. It's very clever and very creative, and he started off doing it in I think 1986 onwards, so it was a long time ago. But yeah, it was that's what he did. It's just very brief summary of it. This is very diagrammatic, and this is just a very brief summary of the process. So